Welcome to third year BSc Botany. I am Amisha Shirodkar-Pednekar, Assistant Professor in the Department of Botany at Parvatibai Chogle College of Arts and Science, Autonomous, Margao. Today we will be seeing the course Cytogenetics and Plant Breeding. The unit that we will be seeing is Quantitative Inheritance, and the module that we are going to study is Quantitative Inheritance of kernel color. In outline, we will be seeing the quantitative inheritance of kernel color in wheat. The learning outcomes: understands quantitative inheritance, explains quantitative inheritance with an example of kernel color in wheat. Introduction: a whole grain or seed of a cereal plant such as corn, wheat, barley, etc. is called as a kernel. Kernel color in wheat is a quantitative trait, and its inheritance was studied by a Swedish geneticist H. Nilsson L. for the first time in 1908. When H. Nilsson L. crossed a certain red strain with a white strain, he observed that in F1 he obtained all the offsprings which were light red in color. And when he self-bred the F1, in F2 he observed that 1/16th of the F2 was like the extreme of the appearance. That means 1/16th was white and the other 1/16th was red. He interpreted these results in terms of two genes, each with a pair of alleles exhibiting cumulative effect. So, if we have to see the cross that was done, this is how it would be. The parents, capital A, capital A, capital B, which would be the darkest red wheat kernel, and small a, small b, which would be the white wheat kernel. Now, here we can understand that capital A and capital B are the two alleles into the picture. Now, when he crossed these two, that is the darkest red and the white kernel, what he got in F1 was an intermediate red color. This intermediate red color had the genotype capital A, capital B, small b. Now, this intermediate red was self-crossed for F2. So, now let me just repeat what I had said earlier so that you understand the next cross better. So, I had said that when the darkest red and the white was crossed in F1, Nilsson L observed that the F1 was all intermediate red and the F2 1/16th was completely white and 1/16th was the dark red, which was like the parents. Now, he interpreted these results in terms of two genes, A and B, each with a pair of alleles exhibiting cumulative effect. So, now if the F1 was interbred, this would be the final result of F2. Let's have a look at this cross. Now, you observe that 1 out of the 16 is capital A, capital B, which is the darkest red, which is like the original parent. And you also observe that 1 out of the 16 is white, which is small a, small b, again like the original parent. Now, the other colors which are observed are, you observe dark red, which has capital A, capital B and small b, which is dark red in color. Then you observe a color which is intermediate red, which has capital A, capital B, small b. Next, you observe that there is a light color which has just one capital alphabet, that is capital A, small b, small b. And then finally, the white. So, if you put these into a ratio, the ratio of the phenotypes becomes 1:4:6:4:1, where 1 is the darkest red, 4 is the dark red, 6 is the intermediate red. Again, the next 4 is the light red, and the last one is the white. Now, if you observe, there are doses of these alleles. So, the darkest red has 4 capital alleles. The dark red has 3 capital alleles. The intermediate has 2 capital alleles. Light has 1 capital allele, and the white has no capital allele. So, this ratio, that is 1:4:6:4:1, has the following: 1 darkest red, 4 dark or dark red, 6 intermediate or medium red. I've put the term medium red also, students, because in case you are referring to some books would call it intermediate red, other books would call it medium red. So, both the terms have been used here. 4 for light or light red and 1 for white. So, if we have to represent this ratio of 1:4:6:4:1 in the form of a graph, it would look like this. First, P1 is representing the parents, that is the darkest red crossed with the white, where in F1 we see that the intermediate or the medium red is obtained. When this medium red is selfed, it is obtained that the ratio of 1:4:6:4:1 is obtained, where 1 is dark red, 4 is red, 6 is the medium red or the intermediate red, 4 again is the light red, and 1 is white. The reference of these colors may be dark red or darkest red depending on which book you are referring to. However, always remember that when all 4 alleles are expressed in the capital form, that is the darkest red which is obtained. Let's come to the quantitative effect. So, each of these contributing alleles, that is A or B, add some red to the phenotype of the kernel color, and that is why when there are 4 capital alleles, that is 2 capital A's and 2 capital B's, it is the darkest red, such that the genotypes of white contain neither of these alleles. We've already seen this in the cross, where there is small a and small b, it is white in color. So, red genotype contains only the capital A and the capital B alleles. These results have already been shown in the images earlier. So, in this case, we got a total of 5 phenotypic classes in the F2. And in these 5 phenotypic classes, what we saw was that each dose of a contributing allele for the pigment production increased the depth of the color. At this stage, one point should be clear, that if it was not quantitative effect and if it was qualitative effect, the ratio would have been 15:1, where 15 colored is to 1 white. However, that is not the case, because the 15 colored ones have varied shades, and those varied shades are because of the quantitative effect. So, here are the phenotypes and the genotypes of the F2. So, if there are 4 capital alleles, that is capital A, capital B, it's the darkest red. If there are 3 capital alleles, that is capital A, capital B and small b, then it is dark or dark red. If there are 2 capital alleles like capital A, capital B, small b, then it is the intermediate or the medium red. And if there is just 1 capital allele like capital A, small b, then it is light or light red. And the absence of any of the capital allele, that is, if there is small a, small b, then it is white in color. So, we can finally say that it is the cumulative effect of the alleles A and B that can be understood in this case. It can therefore be understood that the increase or the decrease of capital A and capital B can cause variable phenotypes in the F2 ratio, which is 1:4:6:4:1. So, in summary, kernel color in wheat is a quantitative trait. Its inheritance was first studied by a Swedish geneticist, Nilsson L., for the first time in 1908. And in this case, when he crossed the red strain with the white strain, it was observed that all the F1 were intermediate in color, and in the F2, approximately 1/16th was like the extreme parents. That means 1/16th was red and the other 1/16th was white. These results were interpreted in the form of two genes, each with a pair of alleles exhibiting cumulative effect. And finally, it was seen that the F2 expressed the phenotypic ratio of 1:4:6:4:1 with various shades of red. These are my references. Thank you.